So we continue talking about populations. And in class today we talked about the idea that populations can change and that each population changes in regarding to some selection mechanism. Not all populations, some selection mechanisms actually keep populations the way they are. If any selection mechanism wasn't actually selecting for any one of these variants of whatever organisms they may be, let's give them bacteria, then the next population would look very similar to this population at all. But in this example, the selection, the environment, natural selection, is actually selecting for only individuals with specific phenotypes, and this could be resistance to specific pathogens or resistance to antibiotics or resistance to heat or resistance to Purell or resistance to soap, for example. And as you see here, after selection occurs, we only have individuals with the selected phenotype. These individuals will no doubt reproduce and produce a population that represents more of what happened after the selection when we've selected for the phenotypes. All right? If we found later on that only specific phenotypes of this population were selected for, like let's say not the high, but the next one, which is this color here, if we found that this resistance level was selected for, then we would find a future population that more represented that. Unfortunately, once a population loses alleles, those alleles would have to come back through either migration or they'd have to come back through some sort of mutation. See, this population here doesn't have these resistance levels, and if they were beneficial in some way, they wouldn't be able to come back unless an organism from another population came in or unless mutation allowed that. So we get into the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is designed to, number one, determine the frequency of two different alleles in a population, really only two alleles, and also to determine if a population, a given population, is changing or not. All right. Now, in order for a population not to change, there can't be any mutation, meaning no new alleles. We can't have any gene flow, which means organisms coming in from other populations changing the allele frequencies. We can't have sexual selection, meaning that one of the phenotypes, P or Q, is being selected for. We also have to have a large population size because any small change in the allele frequencies would offset the balance of what our true allele frequencies would be. And there can't be any natural selection. All right? And what I've just said here is a population will not change if these 5 things can be met. A population will not change unless these 5 things can be met. Now, if we determine that a population is changing, and you'll see how in a second, we can determine that one of these variables is in fact influencing the population. All right? In this example, P is the frequency of the dominant allele, Q is the frequency of the recessive allele, and 2PQ is the occurrence of the heterozygote. As you can see, P squared is the existence of the homozygote dominant, and Q squared is the existence of the homozygote recessive. All right? The frequency of the homozygote dominant plus the frequency of the heterozygote plus the frequency of the homozygote recessive is always going to equal 1 because all the members of your population frequencies will always equal 1. All right? So by understanding this equation, what you're going to see is we can figure out how a population is changing, if it's changing at all or if it's not, and we can determine the allele frequency of a group of organisms based on only knowing one of the frequencies. All right? Now, this can also occur for codominance. It can occur for incomplete dominance as long as there's only 2 alleles present. If we go to our next example here, in this population we have 6 tan bugs and 6 green bugs. If somebody randomly steps on the green bugs, what's actually happening here is the tan bugs are being selected for. Now, this is a random event, but it doesn't change the fact that the tan bugs are being selected for, meaning that the tan bugs allele frequency now is greater compared to the green frequency, and you'll see that in the next population. The next population will show the greater allele frequency of the tan. Now, if we went back here and we saw that only the tan bugs were being stepped on and the green bugs were not, then you would see that this population would represent more of the allele frequency of the green bugs. If we go to this example here, this also represents the Hardy-Weinberg equilibrium. All right, the frequency now this is an example of intermediate inheritance. So as you're seeing here, it doesn't have to be codominance, doesn't have to be dominant recessive. We can do these as with the Hardy-Weinberg equilibrium as long as there are only two alleles. Now we can calculate the frequency of CR by counting the number of times that CR exists in the population. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 out of 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. All right, so 14 out of 20. And then we look at the existence of W. 1, 2, 3, 4, 5, 6 out of 20. All right, this leads us to the frequency of each allele, 0.7 and 0.3. All right, now this is our real population. The Hardy-Weinberg equilibrium is only the expected rate, the expected frequency of that population. For example, if I said how many individuals would you expect of a population to have CR when the frequency is 0.73, what you would do is you would input that 0.7 into this equation. 0.7 times 0.7 equals 0.49. You expect that 0.49, or around 50% of the individuals, would be big CR. Let's see if they are. 1, 2, 3, 4, 5 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And in fact, 50% of them are. All right. You can also do this with the other allele. Would based on Hardy-Weinberg, would we expect, how many individuals would we expect to display the phenotype CW? If we go back and we put 0.3 into here, 0.3 times 0.3, or CW, all right, equals 0.09. All right, so that's about 10%. Right? So if we go back up to here, our 10% of our individuals displaying the recessive phenotype. Let's take a look. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1 out of 10. Or I meant 0.01. So yes, I messed up a little bit before, but yes, our expected ratio is close to the Hardy-Weinberg equilibrium. Now, only 5 of 10 plants leave offspring. Depending on which 5 leave offspring, our next generation is going to be different or the same depending on which offspring come from what parents. Here, we find that our allele frequency changes. All right? Based on this example, we can find that some individuals may or may not be having an advantage. All right? We see the existence of more of the heterozygote of the white plant, the homozygote of the white plants. All right? And we're seeing less of the homozygote with the red plants. We can deduce from this that maybe the homozygote for the white plants is being selected for in this population. Now if we look here, if we go over to the next population, what we're seeing is our allele frequencies are totally different from what this population was and this population. Based on this, we can conclude that either it's random chance that only the red plants mate and produce offspring, which isn't likely, we have or here that the phenotype of being red is selected for,
watermelon.
and that actually these other individuals that are not red are being killed off, die off, or unable to reproduce at some point. So if we look at the frequencies of each allele in a population, we can see that the frequencies are changing. This is what evolution or evolutionary biology tries to determine. All right. Now, when the allele frequencies of a population have changed, we can call that genetic drift. So genetic drift is the idea that the allele frequencies of a specific population are changing. All right. If we look at this example, all right, we have a specific allele frequency for the red and the yellow individuals. All right. If for some reason only the red individuals mate, then they're going to give red offspring, assuming that they don't have a heterozygote in this example. All right. And this shows genetic drift. This bottleneck example is an example that you have a large amount of individuals in a population, only a few of them reproduce, which actually are not representative of the population, and your offspring are again not going to be representative of the population. you can say, and you can actually do this on your own, if the allele frequencies of the red and the yellow allele have changed from this population to this one, and the answer is yes. All right. So there's two different types of genetic drift. One is the bottleneck example, where you reduce the population to a lower number, therefore changing the frequency of the next generation, or something called the founder's effect, where only a few individuals of a population colonize a new area. And in this example, your founder effect, you have 1 in 5 individuals is displaying this red gene here, all right, the marked population. But here, only 1 in, let's say, 20 individuals are displaying that. The fact that 1 out of 5 individuals displays the red gene here, and only 1 out of, let's say, 20 has the red marking here, this actually changes this population. Now this population is going to produce offspring based on this frequency, the frequency of the red and the blue alleles here, not here. So you could imagine your population is now going to have more of the red marked individuals. All right. This is how the founder effect may change the future allele frequencies of a population. All right. The last thing we're going to talk about is the types of selection. All right. There's 3 types of selection: directional, stabilizing, and disruptive selection. All right. In directional selection, one of the phenotypes of an, let's say we're talking about plant size. You have smaller plants, you have mid-sized plants, and you have big plants. Well, in the shaded area, maybe only the big plants are surviving for some reason. That's being selected for. So if only these organisms are surviving, they produce offspring with alleles that match their phenotypes, and over time you're going to have a population that is going to represent the tall. In stabilizing selection, the medium or the intermediate phenotype is being selected for, so you're going to end up with a reinforcement of individuals that are displaying the selected medium phenotype. In disruptive selection, you're going to see organisms, let's say we have short, medium, and tall organisms. If for some reason only the short and only the tall organisms are being selected for, they're going to produce offspring that are going to be either short or tall. Any medium height individuals are going to die off because they're not being selected for in this example. This is called disruptive selection, and leads to species with 2 totally different phenotypes. This type of selection may or may not lead to speciation at one point if these 2 groups become so different that they can no longer reproduce with one another. All right. I hope this gives you a better idea of the Hardy-Weinberg equilibrium and different ways that populations can change. The next thing you got to do is continue on and do some Hardy-Weinberg equilibrium problems. We'll be talking about those problems on Friday, and we'll then be playing our evolution game on Friday and Monday. Look forward to seeing you. If you have any questions, let me know.